A very good day to judges. We are Edith Liu and Tan Li Ying from Raffles Girls' School Secondary, and the title of our project is The Anti-Cancer Properties of Traditional Chinese Medicine. This is an overview of what we will be covering in this presentation. Firstly, cancer is a leading cause of mortality worldwide. This global cancer prevalence has led to the increased demand for cancer treatments of greater effectiveness. In recent years, promising new agents derived from TCM herbs have been in clinical development. Many of these anti-cancer herbs have the unique ability to halt this uncontrolled cancer cell growth, stimulating great interest. This study aims to observe the anti-cancer properties of the 6 chosen herbs and verify if these commonly used cancer-treating TCM herbs indeed exhibit significant anti-cancer properties. These are the 6 herbs used in this experiment. This study utilizes 2 main cancer cell lines, which is the A549 and the H1299 cell line. The A549 cell line originates from the human lung cancer tissue. The H1299 cell line is a human non-small cell lung carcinoma cell line. Both cell lines have been well characterized through several molecular studies, and both are widely used in research and often utilized in cell viability assays. The 6 herbs chosen were extracted using sonication and centrifugation before being diluted into 6 concentrations for testing on the A549 and H1299 cancer cell lines. A colorimetric assay known as the Presto Blue assay was conducted over 3 consecutive days, whereby the Presto Blue cell viability reagent was added directly to each well before 1 hour of incubation. The Presto Blue reagent is reduced by metabolically active cells, providing a quantitative measure of viability and cytotoxicity, whereby higher absorbance values correlate to greater total metabolic activity. From results collated, dose response and survival curves were plotted with the curve-fitting software to obtain the half maximal inhibitory concentration reading of each herb. The IC50 readings represent the extract concentration needed to induce half maximum cell death, indicating how effectively cell proliferation is inhibited. Based on the IC50 readings, the methanol extract of Hidiotis diffusa was the most effective inhibitor of cell proliferation for the A549 cell line, while Scutellaria barbata was the most effective inhibitor for the H1299 cell line. The American National Cancer Institute states the criteria of cytotoxic activity for plant extracts is an IC50 of 30 micrograms per milliliters. However, none of the 6 extracts had IC50 values corresponding to the stated criteria. It can be seen that the A549 yielded more consistent and promising data, with significantly lower IC50 values as compared to the data from H1299. A549 was more susceptible to the cytotoxicity of plant extracts, while H1299 displayed greater resistance against these extracts, generating more fluctuations in data. There are several possible reasons why Hidiotis diffusa and Scutellaria barbata were the most effective inhibitors for the cancer cell lines. Numerous studies, both in vitro and in vivo, have demonstrated the ability of Hidiotis diffusa to effectively suppress cancer cell replication in the colon, lung, breast, and liver. The inhibition of cancer cell proliferation by Scutellaria barbata can be attributed to its precise specificity and high cytotoxicity, causing DNA fragmentation and other potent effects. To briefly cover the limitations and future work of our project, one limitation would be the non-specific nature of our project, which can be solved by conducting a hemolytic assay. Herbs can also be tested for their synergistic properties, since herbs work well in combinations. Other common herbs in cancer treatment should also be included in the study to wider the scope. Furthermore, since effects of herbs are susceptible to variations in sources, many batches of herbs in both granular and dried herb forms should be tested for their anti-cancer properties. Ideally, herbs can be tested in vivo to ascertain if herbs singled out in the studies inhibit cancer cell growth in living organisms. Future work will also include the isolation of active fractions to be used for drug design. The results of this experiment are extremely promising as they show the potential of TCM degradators in combating cancer development. In a bid to discover new treatment techniques and drugs which have less severe side effects and are more effective, this study has shown that TCM products are worth exploring for future drug design and cancer therapies. With careful isolation and purification of the active components within the herbal extracts, new medicines can be produced, tested in vivo, then implemented in clinical testing, and possibly introduced to cancer patients worldwide. In conclusion, this study has shown the anti-cancer effects of selected TCM herbs. Results obtained support existing scientific research in this field and reinforces the knowledge of the anti-cancer properties of TCM herbs. However, results still require further comprehensive studies to be fully substantiated. This marks the end of the presentation. Thank you for your time.